Hello friends. Today we shall talk about the composition of blood, structure and function of hemoglobin and blood groups. The main objectives of today's deliberation are to understand the concept about blood and its composition. We will also try to explain the structure and function of HB, that's hemoglobin. And finally, we will try and know about different blood groups. Let me first of all give you a brief introduction by highlighting the concept of blood and its composition. Blood is an opaque, turbid, sticky fluid connective tissue of reddish color that circulates in the body inside the blood vessels by means of pumping activity of heart. It forms 6 to 10% of the body weight and some 30 to 35% of the extracellular fluid. The study of blood is called hematology. Composition of blood. Blood consists of two components, fluid part called plasma and cells called formed elements. The formed elements include blood cells or corpuscles, that's erythrocytes are red blood corpuscles, leukocytes are white blood corpuscles, and blood platelets or thrombocytes. The plasma and formed elements constitute about 55 and 45% of total blood volume respectively. Now, plasma. The plasma is a faint yellow, slightly alkaline, somewhat viscous fluid. It consists of about 90% water, 1% inorganic salts in true solution, and 7 or 8% proteins in colloidal state. The remaining 1 or 2% of the plasma is formed by food materials, waste products, dissolved gases, regulatory substances, anticoagulants, cholesterol and antibodies. Proteins. The plasma contains a number of proteins: serum albumin, serum globulins, propyridine, prothrombin and fibrinogen. The plasma proteins serve many functions. First, they act as a source of proteins for the tissue cells, which may synthesize their own proteins from them. Second, they serve as acid-base buffers, that is, they maintain pH of the blood by neutralizing acids and bases. Third one, albumin and globulins maintain osmotic pressure of the plasma so that they latter may retain water. Fourth one, plasma proteins transport certain materials in combination with them, like thyroxine is bound to albumin or a specific globulin. And fifth one, some globulins called immunoglobulins form protective proteins termed antibodies in response to entry of foreign agents, the antigens, into the body. Sixth one, prothrombin and fibrinogen play a role in blood clotting. Insulin is combined with globulins. Fatty acids are joined to albumin for transport in the plasma. Now the inorganic salts. The inorganic salts occur in the plasma as ions. Sodium and chloride are the principal cation and anion of the plasma. The anions, bicarbonate and phosphate, and the cations, potassium, magnesium, calcium, iron and manganese, occur in smaller amounts. The inorganic salts are sometimes referred as blood electrolyte. Third one is food materials. The food materials present in the plasma are glucose, amino acids, fatty acids and triglycerides. Their amount depends upon the digestion of food in the alimentary canal. Glucose or blood sugar is an important nutrient which is required all the times by all cells as its respiratory substrate and raw material for carbohydrate synthesis. Now the waste products. The waste products found in the plasma are urea, uric acid, ammonia and creatine. These are removed by the kidneys and their excess causes toxic effect called uremia. Next, dissolved gases. Small amount of oxygen, carbon dioxide and nitrogen are found dissolved in the plasma. Regulatory substances. These include hormones, vitamins and enzymes. Anticoagulant. A natural strong anticoagulant present in the plasma is a heteropolysaccharide named antiprothrombin or heparin. It checks clotting of blood in uninjured blood vessels by preventing the conversion of prothrombin into thrombin. It's produced in the liver. Cholesterol. Liver synthesizes cholesterol and releases it into the blood. It's also absorbed into the blood from the foods such as eggs digested in the intestine. It provides materials to the tissue cells for the synthesis of membrane lipids, vitamin D, steroid hormones and bile salts. Cholesterol normally ranges from 50 to 180 milligrams per 100 ml of blood. Next, erythrocytes are red blood corpuscles. The red blood corpuscles are the most numerous formed elements of the blood. They are the most abundant cells in the human body. A unique feature of the RBCs is the presence of a red oxygen-carrying pigment, the hemoglobin, in their cytoplasm. The shape of RBCs varies in different vertebrate classes. In fish, amphibians, reptiles and birds, they are oval, biconvex and nucleated. In mammals, for example man, they are circular, biconcave, denucleated discs. Their central part is thinner than the margin. Their shape provides flexibility and results in a 20 to 30% increase in surface area as compared to a sapphire. This favors quick diffusion of gases. Human RBCs are smaller than the white blood corpuscles. They are 7 to 8 micrometer in diameter and 2 micrometer thick near the rim. Small size of RBC provides it greater surface area for quick diffusion of oxygen into it. Now we will talk about their number. The RBCs are far more numerous than WBCs. A normal healthy adult man and woman have 5 and 4.5 million RBCs per cubic millimeter of blood respectively. This is called the total RBC count. The instrument that's used for counting of erythrocytes is called hemocytometer. The phenomenon of having exceptionally higher number of erythrocytes is called polycythemia. Lower count of RBCs is known as erythrocytopenia or erythropenia. The RBC count decreases in anemia. Anemia may be caused by loss of blood, destruction of RBCs or faulty formation of blood. The RBCs look yellowish when seen singly and red when viewed in bulk. They impart red color in the blood. The color is due to the presence of a solution of iron containing pigment, hemoglobin, in them. There is about 12 to 16 gram of hemoglobin in every 100 ml of blood. A red blood corpuscle has some 280 million hemoglobin molecules. In the lungs, due to high partial pressure of oxygen, hemoglobin takes up oxygen and changes to bright red oxyhemoglobin. The latter carries 4 oxygen molecules loosely joined to 4 iron ions. Thus, one RBC can carry over a billion oxygen molecules. In the tissues, due to low partial pressure of oxygen, oxyhemoglobin breaks into oxygen and deoxyhemoglobin. In this way, the RBC carry oxygen from the lungs to the tissues. Formation of red blood corpuscles is called erythropoiesis. It occurs in the liver and spleen in the fetus and in the red bone marrow after birth. Proteins and iron are components of hemoglobin, and vitamin B12 and folic acid stimulate erythropoiesis. Deficiency of any of these materials may cause anemia. Human RBCs remain functional in the blood for about 120 days. The worn out RBCs are destroyed by phagocytosis in the blood itself and in the spleen and liver in particular. Their iron is returned to the red bone marrow for reuse in the synthesis of fresh hemoglobin. Their pigment is degraded to yellowish pigment bilirubin, which is excreted in bile. The pale yellow color of the plasma is mainly due to bilirubin. If bilirubin is not excreted fully, the skin and mucous membranes of the person become yellowish, and this disorder is called jaundice. Now we will talk about leukocytes or white blood corpuscles. The white blood corpuscles lack hemoglobin and are colorless. They are rounded or irregular, and nucleated cells can change their shape and are capable of amoeboid movement. This enables them to squeeze out of capillaries into the tissues. This process is called diapedesis. The WBCs are mostly larger than the red blood corpuscles and can range in size from 12 to 20 micrometer. The WBCs are far fewer than the RBCs. Their number varies from 6,000 to 8,000 per cubic millimeter of blood. This number is the total count of WBCs. It may increase or decrease abnormally in certain conditions. Rise in WBC count is called leukocytosis. It is a physiological response to infections, for example, pneumonia, inflammations such as appendicitis, and malignancies such as leukemia or blood cancer. Fall in WBC count is termed as leukopenia and occurs in conditions such as folic acid deficiency, infections of AIDS virus. Formation of leukocytes is called leukopoiesis. It occurs in lymph nodes, spleen, thymus, and red bone marrow. Dead WBCs are phagocytized in blood, liver, and lymph nodes. Leukocytes are broadly divided into two categories: granulocytes and agranulocytes. Granulocytes. These leukocytes contain granules in the cytoplasm and have lobed nucleus, are produced in the red bone marrow. Their formation is called granulopoiesis and have three subtypes: basophils, eosinophils, and neutrophils. First, basophils. These take up basic stains such as methylene blue, are fairly large and have nearly S-shaped nucleus, have few coarse granules. Second, eosinophils. These stain with acidic dyes such as eosin, have bilobed nucleus and abundant coarse granules, contain hydrolytic enzymes and peroxidases which are discharged into the phagosome. The eosinophils have antihistamine properties and their number increase in people with allergic conditions such as asthma and hay fever. The third one is neutrophils. These stain equally with basic and acidic dyes. They are quite large and have many lobed nucleus and abundant fine azurophilic granules, are phagocytic in action. They engulf microbes. Now the agranulocytes. These leukocytes lack granules in the cytoplasm and have non-lobed, rounded or oval nucleus. Agranulocytes are called mononuclear cells. They have two subtypes, monocytes and lymphocytes. Formation of agranulocytes is called agranulopoiesis. Monocytes. These arise in bone marrow, are largest of all types of leukocytes. They have large, sub-rounded or bean-shaped nucleus and a good amount of cytoplasm. They are motile or phagocytic in action and engulf bacteria and cellular debris. Second is lymphocytes. These are about the size of red blood corpuscles, are very large with rounded nucleus and scanty cytoplasm. They secrete antibodies to destroy microbes, and their toxins reject grafts and kill tumor cells. They also help in healing of injuries. There are two main types of lymphocytes, B lymphocytes and T lymphocytes. Platelets. The platelets are rounded, are all disc-like bodies, become stellate in extracted blood, lack hemoglobin. The platelets are the smallest formed elements of the blood, are fewer than the RBCs and more than the WBCs in number. There are about 250,000 platelets in a cubic millimeter of blood. Increase and decrease in the number of platelets is known as thrombocytosis and thrombocytopenia respectively. At the site of injury, the platelets release platelet factor or thromboplastin that helps in blood clotting. Moving on, let's now talk about the structure of hemoglobin. Hemoglobin is the coloring matter of RBC. It's a chromoprotein forming 95% of dry weight of RBC and 30 to 34% of wet weight. Hemoglobin is a conjugated protein which consists of a protein combined with an iron-containing pigment. The protein part is globin and the iron-containing pigment is heme. Hemoglobin is a tetramer composed of 4 globin molecules, 2 alpha globins and 2 beta globins. The alpha globin chain is composed of 141 amino acids and the beta globin chain is composed of 146 amino acids. Each globin chain also contains one heme molecule. The heme molecule is composed of a porphyrin ring which contains 4 pyrrole molecules cyclically linked together and an iron-iron ligand bound in the center. The heme molecule is located between helix E and helix F of the globin protein. The alpha and beta subunits of the globin chain exist in 2 dimers which are bonded together strongly with the iron-iron. And if 2 heme molecules come together in the presence of oxygen, the iron atoms will oxidize and irreversibly bind to the oxygen. The globin chain prevents this irreversible binding by folding the protein around the heme molecule, creating a pocket to isolate the heme molecule from other heme molecules. Therefore, the globin molecules allow the iron atom to form loose bonds with the oxygen and therefore the ability to bind to oxygen and then release it into the tissues without becoming permanently oxidized in the process. Now, it's important to know about its functions. The main function of hemoglobin is the transport of respiratory gases, transport of oxygen. When oxygen binds with hemoglobin, a physical process called oxygenation occurs, resulting in the formation of oxyhemoglobin. The iron remains in ferrous state in this compound. Oxyhemoglobin is an unstable compound and the combination is reversible. That is, the oxygen can be released from this compound. When oxygen is released from oxyhemoglobin, it's called reduced hemoglobin or ferrohemoglobin. Buffer action. Hemoglobin acts as a buffer and plays an important role in acid-base balance. And the third important function is transport of carbon dioxide. When carbon dioxide binds with hemoglobin, carb hemoglobin is formed. It's also an unstable compound and the combination is reversible. That is, the carbon dioxide can be released from this compound. The affinity of hemoglobin for carbon dioxide is 20 times more than that for oxygen. Now we will talk about blood groups. Blood groups are determined by protein molecules present on surface of RBCs. When blood from 2 individuals is mixed, sometimes clumping or agglutination occurs. This clumping is because of the immunological reactions. But why this clumping occurs in some cases and not in other cases remained mystery until the discovery of blood groups. by the Austrian scientist Karl Landsteiner in 1901. He was honored with Nobel Prize in 1930 for his discovery. The immunological reaction that occurs is the antigen-antibody reaction. Landsteiner found two antigens or agglutinogens in RBCs and named them as A antigen and B antigen, and then noticed the corresponding antibodies or agglutinins in the serum called anti-A or alpha antibody and anti-B or beta antibody. However, in the body, a particular antigen and the corresponding antibody can't be present together. If present, it causes clumping of the blood. Accordingly, Landsteiner classified the blood groups. Later, it has become the Landsteiner's law for grouping the blood, which states that if a particular antigen is present in the RBCs, corresponding antibody must be absent in the serum. More than 20 genetically determined blood group systems are known today, but Landsteiner discovered two blood group systems called ABO system and RH system. These are the most important ones that are considered in blood transfusions. First, we will talk about ABO system. Based on the presence or absence of antigen A and antigen B, blood is divided into four groups. First, A group, second, B group, third, AB group, and the fourth one, O group. The blood having antigen A is called A group. This group has beta antibody in the serum. The blood with antigen B and alpha antibody is called B group. If both the antigens are present, the blood group is called AB group, and serum of this group does not contain any antibody. If both antigens are absent, the blood group is called O group, and both alpha and beta antibodies are present in the serum. Determination of the ABO group is also called blood grouping, blood typing, or blood matching. The blood typing is done on the basis of agglutination, which means the collection of separate particles like RBCs into clumps or masses. Agglutination occurs if an antigen is mixed with its corresponding antibody, which is called isoagglutinin. Agglutination occurs when A antigen is mixed with anti-A or when B antigen is mixed with anti-B. To determine the blood group of a person, a suspension of his RBC and testing antisera are required. Suspension of RBC is prepared by mixing blood drops with isotonic saline. The test sera are antiserum A containing anti-A, antiserum B containing anti-B. During blood transfusion, only the compatible blood must be used. The one who gives blood is called the donor, and the one who receives the blood is called recipient. While transfusing, antigen of the donor and the antibody of the recipient are considered. The antibody of the donor and the antigen of the recipient are ignored mostly. Thus, RBC of O group has no antigen, and so agglutination does not occur with any other group of blood. So, O group blood can be given to any blood group persons, and the people of this blood group are called universal donors. The plasma of AB group blood has no antibody. This does not cause agglutination of RBC from any other blood group. The people of AB group can receive blood from any blood group persons. So, people with this blood group are called universal recipients. The blood group of a person depends upon the two genes received from each parent. Gene A and gene B are dominant by themselves and gene O is recessive. Now we will talk about second system, that is RH factor. RH factor is an antigen present in RBC. The antigen was discovered by Landsteiner and Wiener. Since it was first discovered in rhesus monkey, it was named as Rh factor. There are many Rh antigens, but only the capital D is more antigenic. The persons having D antigen are called Rh positive, and those without D antigen are called Rh negative. Rh blood group system is different from ABO blood group system because the antigen D does not have corresponding natural antibody. However, if Rh negative person is exposed for Rh positive blood for the first time, then anti-D is formed in that person. On the other hand, Rh positive person can receive Rh negative blood without the risk of developing complications. Rh factor is an inherited dominant factor. It may be homozygous rhesus positive with capital D or heterozygous rhesus positive with capital D small d. Rhesus negative occurs only with complete absence of capital D, that is with homozygous small d. Now the importance of knowing blood groups. Medically, it's important during blood transfusions and in tissue transplants. Socially, one should know his or her own blood group and become a member of the blood donors club so that he or she can be approached during emergency conditions. Judicially, it's helpful in medico-legal cases to sort out parental dispute. Knowledge of blood groups helps to prevent the complications due to Rh incompatibility and save the child from the disorders like erythroblastosis fetalis. This was all about today's lecture. Hope you enjoyed and understood. Thank you and goodbye.